Good day. We are excited to share our project with you today about the role of Collembola as key indicators of soil health in orchards. I'm Janion Scheepers, the project leader, and I'm based at the University of Cape Town.
Hi, and I'm Jacobs, the MSc student at the Department of Conservation Ecology and Entomology at Stellenbosch University, and the program leader is Mr. Matthew Addison.
Despite the importance of soil for all life on Earth, soil ecosystems and their inhabitants are still understudied. Soil is not only the medium in which trees are planted, as well as a reserve for nutrients and a buffer for environmental changes in the root systems. It is also a habitat for a great diversity of soil biota. These include from the smallest, such as the algae, bacteria, tardigrades, and nematodes, to the mesofauna, such as mites and springtails, and the better-known macrofauna, which includes earthworms, larger insects such as ants and beetles, and arachnids such as spiders. These soil biota play a vital role in maintaining soil structure, integrity, resilience, and health, which in turn also influences the role soils play in the global system. Poorer soils usually contain a lower diversity of soil biota and are susceptible to erosion, compaction, affecting water and air quality. In this study, we hope to use Collembola or springtails as bioindicators of soil quality and resilience in orchards. Collembola are a class of primitive hexapods that are morphologically similar to insects. They are tiny, usually between 2 to 4 millimeters in size. What makes them different from insects is that they are wingless. They have a jumping organ, called a furca, located underneath their bodies, which they use to escape predators, and they also have a ventral tube, which they use for water balance. They also lack a hard exoskeleton, and for this reason they are in direct contact with the soil, making them excellent model organisms to investigate soil health. There are about 9,000 species worldwide, and over 130 species described for South Africa. However, this group is still undersampled in South Africa, with several undescribed species. This group plays an important role in ecosystem functioning, as they are decomposers, but they are also very important prey to many natural predators, such as spiders and predatory mites.
So now I'll be talking about one of the main chapters in the project, Collembola assemblages in orchards, with the aim to compare the differences in Collembola assemblages between orchards using different management strategies. So for methods and materials, we will have 4 replicates per locality or per farm. Half a litre soil and litter samples down to about 10 centimeter soil depth will be taken and transported back to the lab. When the samples are transported back to the lab, they will be extracted via Talgren funnels into 99% ethanol until dry, which usually takes about 7 days, after which the samples within the alcohol will be stored for sorting. Collembola will be identified to morphospecies level using a light microscope. Mites, spiders, and other bycatch will also be stored for any further studies. The same method will be applied throughout the year, as we will sample once every 3 months, which is once per season, to have a better overview of Collembola assemblages throughout the year. So as I've mentioned, samples will be extracted using Talgren funnels, which are lined with mesh and placed over a vial of 99% alcohol. The soil fauna are passively extracted until the soil is completely dry, which usually takes about 7 days. This method is very effective for extracting soil fauna. On the top pictures, you can see our setup of Talgren funnels in the lab, and on the bottom pictures, you can see what the samples look like once they've been extracted in alcohol. On the bottom left, this is the samples before sorting, and the second one is me sorting the Collembola from the bycatch. The third one is an example of a magnified version where you can see there are about 7 species of Collembola present in this image. And the final one is the bycatch, which usually consists of spiders, mites, centipedes, and other soil fauna. With the expected outcomes and impacts, we hope to be able to identify potential Collembola as key indicator species, develop a standard protocol, which is to develop a rapid assessment of the state of agricultural soils by means of using Collembola as indicators, understand Collembola assemblages and diversity in orchards in South Africa, add to Collembola taxonomy, and develop a better understanding of Collembola function.
An ongoing process in this project is to use DNA barcoding, which is a molecular tool which helps us to understand the taxonomic diversity of this understudied group and to identify introduced and indigenous species. This information will help us to develop potential indicator species. The next phase of our project is to understand their functional role in the soil ecosystem in orchards, and this we'll do by using stable isotopes. We would like to thank Hortgrove for funding, the Department of Conservation, Ecology and Entomology at Stellenbosch for their support, and all the lab and field assistants. Thank you.